All right.
Next speaker, Vera Pancaldi from the Spanish National Cancer Research Institute.
Yeah, hello everyone. I'll try to keep it short. I don't want to take time off the wonderful keynote that I'm so looking forward to. So this is a very long title, but basically it's just presenting to you a recent framework that we have developed to try and integrate epigenetic data into 3D chromatin contact networks that in the last but one section were actually quite thoroughly explained. So I'll try to keep it short, but I have a little prelude that might send across the concept without too many technical details. So basically, some people at MIT have started studying the Twitter social network and looking at how they could define happiness for each user based on the type of words that were used in the different tweets of all these different people. And they have discovered that basically happy people go with happy people. So you're more likely to follow on Twitter people that share your level of happiness, or at least the level of happiness that can be inferred by the type of messages you send, the type of tweets. So what they did was actually quantify this using assortativity. And so a very, very simple explanation of assortativity is that you list all the connections in your network, and then you look at the start nodes and end nodes of the connections, and then look at one value, let's say in this case happiness, of the start nodes and the end nodes, and look at their correlation. So what you have here is the plot of these values, so the happiness of user 1 and happiness of user 2 on the y-axis. I guess that's a pointer. And basically you can see a correlation, and therefore you can conclude that happiness is assortative on Twitter networks. So we wanted to know if we could actually apply this to chromatin networks, which is what we're actually interested in. So I'm just going to describe what we did using promoter-centered chromatin interaction networks. This is the promoter capture Hi-C method that was developed by the Fraser lab. And then I'm going to talk about how we used many different epigenetic features to try and project them on this network, and then what we found in terms of biological findings. So basically, the problem with Hi-C, which was actually quite evident from the talks in this room a little while ago, is that you have many, many different contacts of many different regions of the genomes, and because genes are actually a very small part of this very big genome, you don't have that many interactions that involve promoters that to some extent we might be more interested in. So basically what you need is very, very high coverage to be able to see these promoters and their interactions. And the solution that promoter capture Hi-C provides is to add a promoter capture step in the protocol so that you can focus on interactions that involve at least one promoter. So basically we are mapping interactions that either have two promoters, so promoter-promoter interactions, or promoter-other elements interactions. And this is the type of network we get out, where red are promoter elements, blue are other genomic elements, and the edges are colored by PP being red and PO being blue. As you can see, there's a very large main connected component, but also a lot of smaller subnetworks. So how did we actually project chromatin features onto this network? Well, basically, we worked on a set of 78 epigenetic features that include 3 cytosine modifications, 13 histone modifications, and then binding peaks of many different chromatin-related proteins that were basically binarized into 200 base pair windows. And then what we do is for each chromatin fragment that we have in our interaction data set, we look at the abundance of these epigenetic features. So for example, we calculate the proportion of a fragment that has a peak in EZH2, and we basically average this proportion over all the fragments to get a single value of abundance for one feature in the network. And then we calculate for each one of these features the assortativity on the network. And this is the value I was mentioning before. So it tells you whether two chromatin fragments that interact with each other have a similar value of this feature. And this is, it depends on the abundance of this feature. Obviously, if you don't have any fragments with that feature, you won't have assortativity. But it's really not just that. It actually tells you about the topology of the network and how that is related to the presence of the feature, or at least associated. So this is just a depiction of the promoter-centered network in mouse embryonic stem cells. In fact, here I'm just reproducing the promoter-promoter network just to show you. And the statistics, I guess you can't see that, but there's basically chromatin fragments have a size that is on average about 5 kb. And there's about 55,000 nodes and 90,000 more, 19,425 are promoters. And again, we have about 20,500 edges that connect to promoters. Also, I'd like to highlight that the recent data that was published with promoter capture Hi-C was actually processed with a different tool compared to what I've used, because those data were processed with something called Gothic, which is a pre-processing algorithm that goes from a map of interactions of chromatin fragments that you can get in a Hi-C experiment or promoter capture Hi-C experiment. Then you have to basically select the statistically significant interactions, and this Chicago method that should be out soon and is already available as preprint is actually processing the data in a slightly different way compared to the published data set. So the data set is published but processed in a different way. And basically, this is what I want to show you, that we have calculated assortativity and compared it with abundance for all these different features. Each dot on this plot is actually one of our features, and they're basically color-coded by type. So this sort of pink nodes are actually Polycomb-related features. Red points are actually the RNA polymerase complex. And then we have other types of protein and histone modifications. So what I want to show you here is that the Polycomb group of proteins and associated histone marks actually shows a very high assortativity level, even though it doesn't actually have a high abundance. So these are actually not very widespread elements on the genome, but they seem to be very related to the fact that there are interactions with the fragments that have that mark. And the other thing I want to show is the fact that the polymerase features, and there are 5 of these, and I'll get back to that later, also have a pretty high assortativity level compared to their abundance. And basically, the fact that Polycomb is involved in holding together the spatial structure of, the spatial network of interactions of chromatin is actually not entirely new. And actually, this paper was coming out this summer where they actually knocked down some of the Polycomb members and show that knocking down the elements destroys the network that holds together the contacts, at least at the Hox genes subcluster. But basically, this RNA polymerase 2 also shows some important function that I will discuss further. So the other thing that we wanted to do is that so far we've looked at all the contacts independent of whether they were promoter-promoter or promoter-other end. And what we wanted to do was actually look at differences between the two. So in each contact network, we separate these two subnetworks, PP and PO. And here what I'm showing is on the y-axis, the promoter-other end assortativity, whereas on the x-axis, we have assortativity in the promoter-promoter contacts. And this is the same set of features that I showed before with the same color coding. So what you can notice, although you cannot read it, but basically the Polycomb is actually sitting here on the diagonal, which suggests that the high value of assortativity of Polycomb features is actually equally important in the promoter-promoter contacts as much as in the promoter-other end contacts. Whereas if we look at the polymerase features, we see a very interesting fact, that basically in promoter-promoter contacts, assortativity is the same for all these different RNA polymerase variants, but it actually changes in the promoter-other end elements. And I will come back to this very soon. The other thing we can notice is that there are features that are actually positively assortative in the promoter-promoter context, and this means that features that have similar values of these, fragments that have similar values of these features tend to be close to each other. But when you look at the promoter-other end context, they actually have negative assortativity. And this means that when you have a pair of these fragments, one will have the feature and the other one will almost always not have it. It's a negative correlation. So we see, for example, H3K4ME1 in this category, and this makes total sense because basically in the promoter-promoter fragment contact, you imagine that these are two promoters. They are likely to be enriched for this mark, which is typically promoter mark. But when you look at a promoter with another end element, you do not expect a promoter mark in the other end, and this is why you have negative values. Okay, so the thing that I've been postponing to explain so far is basically what we did with the 5 different types or variants of RNA polymerase that we had in our feature list. So basically, these ChIP-seq data that obviously we didn't collect ourselves actually came with 5 different types of features for polymerase because the antibodies used in these experiments were actually basically specific to different variants. So the two forms called POL2 and RNAP, well, 8WG16 basically, they are unspecific. They recognize polymerase in any of its variants, whereas we also have ChIP-seq signals for polymerase in its active form or its elongating form. So I've used color coding to show that basically this is unspecific, and then as you move towards redder and redder colors, you get a more active polymerase, and the properly red color shows elongating polymerase. So what we noticed on the previous plot, which I've actually reproduced here in a bar plot to show it better, is that whereas in the promoter-promoter contacts, the assortativity of the different variants of polymerase 2 is constant, when you look at the promoter other end contacts, you see very big differences in the assortativity of the different variants, with the elongating form having the highest assortativity. In fact, what you see is that the assortativity of the non-specific variants is actually low. It's not like this one is higher, it's that this one has fallen. And basically, we have also looked at this, trying to separate between promoter other element contacts that actually connect promoters to active enhancers. And to do this, we basically looked at the features in the other elements and looked for the typical marks of enhancer activation. So, for example, we looked at HTK4, that should be ME1, with HTK27 acetylation, or with HTK27ME3 to have poised enhancers. And basically, we see that the increase in assortativity of the more active forms of polymerase is happening in the active enhancer case, but not in the poised enhancer case. And this kind of strongly suggests that there is a role of the elongating form of polymerase in establishing the activity of the enhancer, or at least to suggest that active enhancers will have this elongating form of polymerase. The other thing I want to mention is that although I've shown that there is a dependence on abundance, these relationships are not due to the abundance. They are actually due to the topological distribution of the feature on the network and not simply by how abundant these features are. Basically, what I wanted to show you with a simple example is that we have generated this methodology that we believe could be applied in many different cases. It can certainly be used for different data sets. In fact, we have applied it to a parallel data set, which is called, well, it's by Salen et al. And basically, they have done a similar promoter capture Hi-C protocol, but using a different enzyme to do the restriction step. And so you get much smaller fragments and a higher resolution picture. And we have actually reproduced our conclusions, obviously with a few differences, because clearly the different size of the fragments make a difference. But we can reproduce our results in a similar data set. And we have also reproduced all these findings, although with many, many more specific differences between datasets, using ChIA-PET datasets that were recently published. We've done it with a cohesin ChIA-PET and with the polymerase ChIA-PET dataset. So if you're interested in this, I can tell you more about it. But basically, we are quite confident that this method is telling something interesting, because there are recent papers and recent findings that are actually supporting our finding that basically they are identifying contacts between distal elements, or at least regulatory elements, promoters, and gene bodies of the genes. And this is basically pointing to the fact that the elongation step of the transcription might actually be very important in the enhancing action. The other thing that I cannot really comment so much about, because we're still investigating, is that we see many, many contacts between promoters and bodies of other genes. So we can expect that if we have the elongating polymerase at the promoter and then the gene is being elongated, there will be a contact between the promoter and the elongating part of the gene. In fact, there's a recent paper by the Blobel group which is showing these contacts by 3C. But basically, we are interested in knowing when we can see a contact between the promoter and the gene body of a different gene, what does that mean? Could it be that these genes are being transcribed somehow locally in the nucleus, and so we would see the contact between promoters and promoters, but because of the low coverage of these methods, we're losing that? So these are open questions that I'm very happy to discuss. And this is just to conclude. So we have applied a metric from social networks to look at epigenetic features projected onto 3D contact networks. And it has allowed us to estimate which features are important. And we recapitulated the role of Polycomb and suggested important roles for the elongating form of polymerase. And then importantly, this method also allows to compare chromatin interaction data sets between each others. Like I showed the comparison between the two different subnetworks. We can also look at two different data sets, one against the other. And hopefully this addresses some of the problems that this field is currently having in terms of interpretation of these very large and complex data sets. And finally, this method takes into account the nectar topology. Right, so these are acknowledgements. This is my group. This is Alfonso Valencia, so I'd like to thank him. These are my co-workers that have collaborated on this work. And I also want to thank the Babraham Institute with Peter Fraser and Mikhail Spivakov, who have been really key in providing data and the processing of the PCHIC data. And this is funding, and if you have any questions, I'll be happy to take them. Thanks.
Thanks so much, Vera. So we're slightly behind schedule, but since the other session is 4, so
Lucky.
time for questions.
So I was just wondering about 5-methylcytosine. I couldn't find it on the associativity plot. I saw 5-hydroxymethylcytosine and formal cytosine.
We have them.
So yeah, but in the plot, why was it actually, or at least it looked like it was very far away from the cluster of 5-HMC and 5-FC? I was wondering if you could say why that might be.
Well, I don't think they appear at the same, they don't have the same assortativity. In fact, we're actually looking very closely at the differences between 5MC, HMC, and FC, because there are interesting patterns of what happens when you look at the assortativity in the promoter-promoter context and promoter-other-end context.
Right, I mean, what I thought was particularly interesting is that 5MC seemed to be very different, but 5-HMC was very similar to 5-FC, and I guess it's not explained by abundance because 5-FC is 1 to 2 orders of magnitude less abundant than 5-HMC.
No, it's not abundance. It's assortativity. I mean, basically, it's where they are on the network.
Right.
We can talk later about that.
Sure.
We're very interested in that, actually.
Thanks.